ペール博士ありがとうございました。共生の科学と技術分野で、受賞されましたアシュトン博士に、熱帯林の半世紀、その危機と神秘、生物多様性の持続的利用を求めてというテーマでご講演をいただきます。Dr. Ashton, please.
Good afternoon. Ladies and gentlemen, and welcome, friends. It's really most exciting to see so many friends in the audience this afternoon and to be with you here in this marvelous country of yours again. Thanks, I suppose, to one or two of you here, too. I'm delighted. I'm also very honored. Last month, I celebrated my 50th year of forest research in the Asian tropics. In that period, that 50 years, those forests have experienced greater change than in the previous 15,000, and even in the fastnesses where their species survived during our northern ice age in the previous 10 million years. In 1957, I had been appointed forest botanist in good Sultan Omar's government in Brunei Darussalam, my arrival in Asia. Brunei, blessed with oil, had retained 9/10 of its land under aboriginal forest. Its peat swamp forests, though deriving their nutrients solely from rain, were the tallest in the tropics. Now, 50 years later, it's hard to find rainforest trees as magnificent as the dipterocarps that then clothed Brunei's lowlands. At that time, virgin forest, hunted but never cut, covered 80% of Borneo and not much less of Sumatra and Peninsular Malaysia, while well-managed rainforests extended through Burma and South Asia. Now, virgin forest is hard to find outside the few preserves and some mountaintops, and is even going within several of these. It is being converted to temporary agriculture by desperate immigrants inexperienced with these infertile lands, which is eventually abandoned, or now, increasingly in the lowlands, to commercial oil palm plantations. Should the world be concerned? I put that question to you. Should we worry about this? Tropical forests store carbon in biomass, though no more than many temperate forests. Their soil generally stores less carbon than temperate forest soil, though the peat swamps that are unique to Far Eastern forests have accumulated soil organic matter at unprecedented rates. Their burning, often intentional in years of drought, has been estimated to release as much carbon into the atmosphere as all the industrialized nations together over the same periods. The forests of the tropics may play a mediating role in global climate, particularly those of the African and South American continents, but it is their biological diversity which distinguishes tropical forests from all other terrestrial vegetation. More than half the species on Earth are confined to tropical forests, and more than 3/4 of these are confined to the rainforests, mostly in the equatorial regions. And again, here, biodiversity is confined to the lowland rainforest, that is below 700 meters, the very areas under greatest pressure for conversion to agriculture. Fundamentally, biological diversity represents genetic diversity, the diversity of gene sequences found in nature. As a more tractable measure, biologists use species diversity as a reasonable proxy measure of biodiversity. Species diversity is overwhelmingly concentrated among insects and microorganisms, which, directly or indirectly, depend on plants, the primary producers of organic mass. In tropical forests, the living mass is in trees, And there many thousands of tree species have evolved over millions of years, a vast pharmacopoeia of defensive chemicals which have spurred co-evolution of their insect and microbe enemies. Does it matter if we lose the majority of these in one human generation? Biodiversity represents a library of genetic information. As in a great library, corridors may only be visited once in a decade, a century. But those visits sometimes lead to major discoveries. The loss of the great library at Alexandria in Egypt in medieval times put back the advancement of European culture by centuries. We cannot know how the genetic information so overwhelmingly curated in the lowland tropical rainforest will benefit life generally. The chemical diversity it presides over will certainly assist in plant and animal disease control, besides furthering pharmaceutical discovery, while understanding the delicate balance of species which harbor it can assist in the design and management of artificial ecosystems. When naturalists first entered the forest of Borneo, the nature of the forces which appear to sustain the extraordinary equilibrium that is rainforest species diversity could only be subject to speculation. The great colonial botanists of the Far Eastern rainforest, Kees van Steenis of Bogor and Leiden, and John Corner at Singapore and Cambridge, perplexed by the forest's unfathomable diversity and apparent lack of spatial pattern among a tree species, surmised that there is no predictable pattern of species association, either in time or in space. My friend and colleague Stephen Hubble, then at Princeton, has recently elegantly demonstrated in his unified neutral theory of biodiversity that sustainment of tree species diversity by random processes, notably the limited dispersal of seed from the scattered parent trees, is indeed theoretically possible. That would imply that the species are ecologically complementary to one another, so it has immense implications for Darwinian natural selection and evolution. Do rainforest tree species obey the laws of natural selection, leading to specialization? Or are their communities a random mix of ecologically complementary species, which changes over time and in space, mediated by these trees' limited seed dispersal? Foresters had earlier noticed that seedlings and larger juveniles do indeed cluster around the trunk of parent trees in tropical rainforests, as they do in temperate forests, owing to limited seed dispersal. And here we see a diagram of one dipterocarp species, one rainforest emergent tree species, Macroptera, and the scale here, as you see, is 1 by half a kilometer. And I note this because it's the one example I'll be showing of the global network that now exists of very large plots for tree species demographic research in tropical rainforest, the CTFS, that I will return to. Hubble's work serves as a rigorous null hypothesis against which to test for evidence of community structure caused by tree species ecological specialization. If rainforest trees, like our temperate forest trees, are habitat specialists, their distribution and response to physical conditions of soil and light are predictable. Therefore, habitats will support distinct species associations, and site-specific management for specific objectives is to a significant extent possible. But if their species are ecologically equivalent, their behavior cannot be predictable. Then, conservation management can only depend on a law of island biogeography, which states that there is a relationship between the area, species occupy and the number of species that area can contain. Therefore, the number of species a forest can support will decline as its area is reduced. For larger mammals and birds, it has been found that the reduction of a natural area by 90% eventually leads to the loss of half of its species. I was initiated into rainforest ecology in Borneo with but a rudimentary herbarium, museum, plant museum, and library. Instead, I had the companionship of 4 young and enthusiastic Iban Dayak woodsmen and skilled climbers for collecting the necessary flowering specimens high above in the canopy. Marvelously knowledgeable, they taught me how to perceive forest diversity through their eyes, and I thereby gained a deep understanding of the forest which no book could impart. There were no inland roads in those days. My mission then was to document, name, and classify the species of timber trees. Once I had learned the commoner species, to my great surprise, I began to realize that different hill ranges and different parts of the topography bore different suites of species associations, and that these associations repeated themselves in different localities where similar habitats were re-encountered. I noticed, in particular, that the species on the coastal hills of young sandstone geology supported one group of overlapping species association, whereas the older and predominantly shale hills inland supported another. With my companions and the assistance of the Forest Department, I laid out and censused the trees in 50 plots on the ridges and slopes of the inland hills at one locality, and another 50 at a locality on the coastal hills. These plots were found to contain almost 1,000 tree species. That's to compare with some 500 known from North America north of the Mexican border. And I was at a loss to know how to test, by their analyses, whether detectable patterns of species composition existed among such a complex data set. But by an amazing stroke of good fortune, J.R. Bray and J.T. Curtis, studying forests in North America at that time, published a robust and informative method for analyzing species patterns among forest community samples and testing for the relationship of these patterns with habitat. The method involves comparison of all plot pairs using coefficients based on the species they contain. The similarity of species composition between plots expressed in these coefficients was then depicted graphically, the distance between the plots in 2 or more dimensions reflecting their similarity. Working for a month, night and day, cranking a hand-operated facet calculator before the days of readily accessible computers, my plots formed some clumps, but many were scattered, as you can see here. But when I superimposed the topography, just as when a photograph begins to develop in the hypo, a marvelous picture emerged embodying the patterns of species within the landscape that I had sensed in my wanderings. Not working? Okay. My student Savitri Gunatilaka, now at the University of Peradeniya, found similar patterns during her dissertation research in Sri Lanka rainforests. We had proven for the first time that rainforest species do indeed associate together in specific habitats at each locality. But it was still unclear whether the large difference in tree species between the coastal sandy and inland clay habitats was due to their habitat differences, or, on the other hand, to their 100-kilometer distance apart, and to the random exigencies of their limited seed dispersal, therefore. Later, with colleagues in the neighboring Sarawak, Malaysia Forest Department, I extended plots throughout the lowland dipterocarp forests there, set it up in 13 localities of diverse geology, and up to 500 kilometers apart. With the help of a biometrician and a former student, Matthew Potts, these were analyzed using a more recent, precise method in this diagram. Here, the plots are divided sequentially from a common pool at the right. Here's the common pool. They are mostly grouped by their locality, these groupings, suggesting some influence of limited seed dispersal. But these groupings also correlate with soil factors, which are indicated in these boxes. Moreover, the primary division here, into two major divisions, one there and one here, again distinguishes between plots on less fertile sandy soils in the upper first division and those on more fertile clay soils down below. The groupings are divided in this diagram into 4 geographical regions indicated by 4 colors, which you can see there. With the exception of the blue group from the southwest at the top of the diagram, the geographical provenances are scattered through the diagram, implying that the influence of soils on species distributions overrides their distance apart, even at 500 kilometers, and by inference, dispersal constraints, even at regional scale. Potts statistically confirmed the dominance of soils factors over distance. These results confirm, therefore, that habitat-specific management is possible in tropical rainforest. These results also provide the first rigorous proof that hyperdiverse tropical rainforest species specialize in relation at least to soil. Each habitat is found to contain many species, however, estimated from these and later studies in Peninsula Malaysia and Borneo to be about 500 to 700 tree species in a single soil, a single topographic unit. Does community species diversity itself vary in predictable ways that can be correlated with their habitats? We found, using the same Sarawak plots, that though diversity within small samples varied by other factors as well, as can be seen in this diagram, it did show a marked trend with soil diversity here measured as magnesium concentration, from low levels on less fertile sandy soils at the left, rapidly rising with increasing fertility, to a peak on soils still of quite low fertility, and then, to our surprise, actually declining in diversity as soil fertility increases further. At each level of fertility, the forests bear a distinct, albeit overlapping array of species. All must be conserved if all species are to be retained for the future. There appear to be several possible causes for this pattern of diversity. David Tillman at the University of Minnesota has hypothesized that as soil fertility increases, the number of ways in which species can specialize in their use of the panoply of soil's nutrients also increases. But eventually, nutrient levels reach saturation, after which a few fast-growing species outgrow and thereby outshade the rest, suppressing species richness. Alternatively, species diversity is mediated by variation in the impact of drought on tree canopy tree mortality. On sandy, drought-prone, as well as infertile soils, at the top here, canopy is low, and emergent species suffer too frequent death from catastrophic drought to persist, and the forest is short. On compact clay soils of intermediate fertility in the center, emergent trees experience less frequent selective drought mortality, thus providing gaps, but not entirely disappearing, the tall emergent canopy, not entirely disappearing. But other shorter canopy species survive between them, thereby increasing the overall species diversity. On well-watered clay loams, at the bottom there, emergents seldom, perhaps never experience severe drought, and the canopy becomes almost continuous and dense, suppressing the species diversity beneath, somewhat in the way that Tillman predicted. In effect, both explanations appear valid according to the particular habitat sequences. To understand the biological causes behind the patterns, we needed to seek evidence of specialization among the species themselves. A characteristic of hyperdiverse rainforest is the co-occurrence within one habitat of series of species closely related, sharing the same genus. Such series occur in Japanese forests, for instance, among the oaks in the evergreen forests, and among the maples in the deciduous to the north. These closely similar species are the most likely, on the one hand, to be ecologically complementary, but on the other, to be the most stringent, under the most stringent competition. Do closely related species differ ecologically, is our next question. To find the answer requires a combination of carefully monitored population samples of related species combined with experimental transplant and competition studies. Populations of all but the commonest species are widely scattered in these hyperdiverse communities, therefore requiring very large samples monitored over very long time periods. Therefore, a network of very large plots was required. This could also assume the role of global monitoring system for evaluating the influence of changes in the climate and atmosphere on the performance and competitive relationships in hyperdiverse terrestrial ecosystems. Stephen Hubble had in 1980 developed protocols for a plot of 50 hectares, such as the one I showed you earlier, in seasonal rainforest at the experimental station of the Smithsonian Tropical Research Institute, STRI, on Barrocola Island in the Panama Canal. With support from the US National Science Foundation, all 250,000 trees exceeding 1 centimeter diameter at 1.5 meters above the ground were mapped, measured, tagged, and identified with the intent to recensus at 5-year intervals. He presented his analysis, which purported to support his neutral hypothesis and implied high ecological similarity among the species, at a meeting in 1982 at the British Ecological Society at Leeds in England. I was there. After listening to his presentation with skepticism, I invited him to a pub for some beer, where we launched a vigorous argument. Very good place for scientific discussion. It resulted in our agreement in jointly seeking funding for a replicate plot in Asia. The Forest Research Institute, FRIM, of Malaysia, joined us, and the plot was established by them at their Paso Research Forest in 1985. The large plot approach to demographic and ecological research on rainforest trees attracted the interest of several scientists and friends in tropical Asia. The idea of a network of plots representative of species-rich rainforest community types, censused in a uniform, therefore rigorously comparable manner, and large enough to capture adequate population samples of a majority of their species for demographic research, had arisen. Such a plot network, analogous to the global networks of plots for testing crop varieties, had never been established on such a scale in terrestrial natural ecosystems. A plan for tropical Asia was discussed at a conference in Bangkok in 1989, and thanks to funding from several sources, was initiated the following year. Meanwhile, discussions with Hubble and STRI were started for plots in the neotropics and Africa. And in 1991, Steve, Thomas Lovejoy of the Smithsonian, and I met at STRI, and the Center for Tropical Forest Science, CTFS, an organ of STRI, whose Asia chapter was to be administered from Harvard's Arnold Arboretum, where I was working, was inaugurated to coordinate what was quickly becoming a complex international partnership. Natural scientists in tropical Asia had become increasingly focused on research within their own frontiers, with few opportunities to collaborate internationally, let alone visit forests throughout their region. Thus, from CTFS has arisen a regional and indeed shortly a global community of forest scholars exchanging experience, sharing data, and yearly visiting unfamiliar sites and unfamiliar problems during our yearly workshops. It is particularly gratifying that Japanese researchers, both scientists and students, have predominated among collaborators from outside the region. They continue to contribute major advances to the research efforts. Japanese funding, notably from the Mombusha and from Nice, the National Institute for Environmental Studies, has significantly assured the success and continuation of the program in Asia. The immense training opportunities provided by the diversity of sites and species represented by the millions of trees now under observation promises a renaissance of tropical field biology and forest research. Results are already being applied in research towards reforestation using indigenous species of economic value in Sri Lanka and also in Panama, while this year resource economists and forest ecologists will collaborate with CTFS to start addressing the economics and ecological methodology for sustaining biodiversity in indigenous timber production forests under the auspices, once again, of FRIM in Malaysia. The central quest of CTFS research remains to better understand how hundreds of species coexist in tropical rainforest communities. Studies have focused on co-occurring closely related species. In every case, the species investigated have proven, in fact, to be ecologically distinct, inferring divergent evolution through natural selection. Thus, our former student, Sean Thomas, now at the University of Toronto, showed that co-occurring trees in the subcanopy genus Bacauria at the Paso, Malaysia plot start reproducing at different sizes and reach different ultimate stature, consistent with the theoretical prediction of Takashi Koyama, Hokkaido University, that species can coexist by diversification of their heights at maturity. Toshihiro Yamada, now at the Prefectural University of Kumamoto, likewise showed that species of the canopy genus Scaphium differ in ecological range relative to both topography and soil, and where two species do occur in the same microhabitat, they reach different parts of the canopy at maturity. Our son Mark, now professing silviculture at Yale University, examined the comparative physiological ecology and anatomy of a co-occurring species of the Dipterocarp genus Shoria in the Sinharaja, Sri Lanka CTF forest site, in collaboration with Savitri and Nimal Gunatilaka there. These species occur in overlapping ranges on different elements of the topography, as you can see in this photograph. He found that canopy tree mortality, hence canopy structure, from ridge to valley, there mirrored the gradient observed from dry to moist soils in Sarawak. Each species along his gradient differed markedly in response to soil water and light. And here you can see along those two gradients, light horizontal. This is the light gradient and this is the water gradient in experimental plots. Perhaps least to be expected, my former student Stuart Davies, now director of the global CTFS program, investigated 12 species of the pioneer tree genus Macaranga, which occur at the Lambia Sarawak CTFS plot and site. By demographic and experimental study, he showed that each species was distributed differentially in relation to crown exposure to light. 11 different species, each with its own distinct difference in density in relation to crown exposure, difference in numbers in relation to crown exposure, that is light. And they differed also in growth response to soil factors, while each started to reproduce at different heights, reached different final statures, and had different longevity. Nevertheless, tropical rainforest communities contain far too many tree species for any possibility of consistent physical competitive interaction, either in space or over time. So these results are difficult to explain in terms of simple physical competition. Further, although related species differ ecologically, implying that each may occupy a different niche within the forest structure, as we have seen, the species series of different genera appear to overlap ecologically. Instead, motile organisms may act as the mediators of much of this competition between trees of different species and genera. To examine these interactions, it is necessary to ascend into the canopy and to coerce our students to brachiate among the distant foliage above. The major advances in our understanding of the insects that pollinate rainforest trees and the birds and mammals that disperse their seeds are thanks to the extraordinary leadership, innovation, enterprise of the late Temiji Inoue, University of Kyoto, his students working at the Lambia CTFS site, notably the late Kuniyasu Momose, and Shoko Sakai, I'm very glad to see here with us this evening, and her colleague Takakazu Yumoto, all of Kyoto University. Until recently, the pollinators of great dipterocarp trees which dominate the canopy of Far Eastern hyperdiverse rainforests were unknown. They are now known to include bees, moths, and many other insects in different genera, often shared among tree species within a genus. Sakai found chrysomelid beetles on the flowers of Shoria species at Lambia. At Paso, Chan Hung Tak, the person in the picture here in the chair, and Simithiri Apana, then students at the universities of Malaya and with me at Aberdeen University, Scotland, examined the reproductive biology of 6 co-occurring emergent species there at Paso. Canopy dipterocarps in the aseasonal wet forests of the Far East flower as a family approximately every 5 years, apparently in response to a drop in minimum night temperature associated with the El Niño climatic cycle. Individual species populations flower over a short period not exceeding 3 weeks, during which each tree may open up to 4 million flowers. Where do the pollinators come from? 4 million flowers per tree, millions of trees in the landscape, once every 5 years. These Malaysian students found the buds of the dipterocarps, of the Shorias, to be inhabited by thrips, Thysanoptera, which are abundant pollinators of temperate climate flowers. This little creature here is about 3 millimeters long. Their work revealed a wonderful story. By monitoring canopy insects using traps and by breeding the thrips in captivity, they found that thrip numbers greatly increased prior to Shoria flowering, and that they lay their eggs in the young Shoria buds, where the larvae may mature over 3 life cycles before the flowers open, enormously increasing their numbers. The thrip larvae feed on the petals, but do not harm the maturing anthers, whereas the mature thrips suck out the pollen grains, but cannot remove the sticky grains that adhere to their backs. The flowers, with twisted adhering petals, open in the evening and gyrate to the forest for the following morning, with many thrips on board. These then fly erratically with sticky pollen attached to their backs like a diver's oxygen cylinder to the canopy, where they may reach flowers on a different tree from that from which they originated. Unlike in wind-borne pollen, the thrips can row through the air with their oar-like wings, alighting directionally on a nearby open flower. Avner and Chan carefully monitored the flowering times of population samples of their 6 shoria. They found that they flower in overlapping sequence, but that for all but one species, there is a period when one alone is in flower. That period is marked by the thick line, the thick stripe there. If the length of population flowering periods is taken into account, it was found that flowering times significantly departed from random expectation. That implies that competition for the pollinators or pollution of stigmas by foreign pollen, thereby reducing fecundity, has led through natural selection to different flowering times. This remains, as far as I'm aware, the only rigorous evidence, albeit inferential, for differential selection among co-occurring related rainforest tree species in response to competition for the services of a motile organism. It remained to be resolved how rainforest trees come to be confined to specific soils or topographic elements. Sabrina Russo, working with Stuart Davis and with myself and the Lambia plot data, found unexpected patterns of growth and mortality among the trees. Here, each column in each histogram it represents mean growth of species found mainly, but not exclusively, on each of 4 soils of increasing fertility and increasing clay content from left to right, with generalists in the 5th column on the right. She found, surprisingly, that the rank order of growth response, down here, this set of histograms, each on different soils, as can be shown there, in the 4 histograms to the left, did not change among the 4 groups of soil specialist species when all were grown on each of the 4 soils. The rank preference for those soils remained the same. But she also found in the right-hand histograms that the specialists on the least fertile soils, that's these ones, in the white column there, had markedly lower mortality, as you can see, during the period of measurement. Together, these results may imply that some organism differentially kills specialists of more fertile soils when they establish on soils below their preferred fertility. Akira Ito, Osaka City University, has carried out exemplary comparative research on the 2 species of the emergent Dipterocarp Dryobalanops at Lambia, confined respectively to fertile clay loam and infertile sandy soils there. In seed transplant studies, he found that germinating seeds of the species of fertile soils differentially suffer taproot herbivory when placed on the infertile soil. And similar results have now been obtained in seedling transplant experiment in South America. Herbivory, and more particularly host-specific pathogens, may eventually provide the principal explanation how the extraordinary diversity of rainforest tree communities, which reach their zenith in equable, humid, hot climates, may be maintained. In these windless climates, fungal spores have limited dispersal. Like children in a classroom, most neighboring individuals of a tree species may be prone to infection, but those which disperse further away, in a separate classroom, as it were, may escape. Thereby, space is created in which other species resistant to the particular pathogen can survive. A striking example is provided, again, in the peat swamp forests of Sarawak, dominated by that single emergent Dipterocarp, Shoria albida, shown in my first picture today. These swamps are young in geological terms, about 5,000 years old. the dominant species is being killed in vast patches. Here you see in 1957 or 47, and here again, 15 years later, a vast patch of dead trees in the canopy. Apparently by the larva of a moth whose female is wingless. This mass death represents, it seems, the first stage in the conversion of the peat swamp canopy of a single species eventually into a multiple species association. We have seen, in the case of exotic crops introduced free of pests and pathogens, such as rubber and oil palm in Southeast Asia, that escape from predators can be sustained for sufficient time to ensure their financial success. But there are grave implications from these observations for current attempts to grow indigenous tree species in greatly reduced species mixtures, even monocultures. These results also explain why clumping is so much more marked among seedlings and saplings than among mature trees. But obviously, some individuals survive. Are pollen and seed of rainforest trees actually sometimes dispersed much further than they appear to be? Seeds of tropical trees, unlike pollen, are mostly dispersed by vertebrates. They may occasionally carry the seed outside or inside the gut quite long distances. Vertebrates are leading pollinators too of some rainforest tree species. A striking example, seen in this wayside stall in Malaysia, is the pollinators of wild bananas, and also the bean-producing forest tree. You see these beans here? The bean-producing forest tree Parkia, and the celebrated durian fruit of the Asian tropics. The pollinator is a fruit bat, which feed exclusively on nectar and pollen. The bat pollinator of the durian and Parkia trees is this one, Eonyctra spelea, which forages in flocks and roosts in caves. A student of mine, Anthony Start at Aberdeen, was able to follow its feeding choices by weekly collecting the guano beneath the bat's roosts in the cave and identifying the distinctive empty pollen cases that pass through the bat's gut. He found that these bats also fed on the flowers of two species of coastal mangrove trees of the genus Soneratia, mapped here, whose distribution in estuaries and along the coast are easy to map. From this, he could infer that these bats commute every night up to 50 kilometers each way, solely on a diet of nectar and pollen. Bats then have lessons for us commuters, for our carbon emissions and global warming. More direct genetic evidence is now appearing which indicates that seed dispersal and gene exchange can occur over much longer distances among rainforest trees whose seed dispersal is mostly limited. Nevertheless, there is other recent evidence that the power of natural selection for specific habitats overrides the generalizing force of distant dispersal. What are the implications of these discoveries, fascinating to the naturalist, but less obviously so for the timber merchant or the policymaker? Plants, unlike most animals, cannot move during their lifetimes. They are more habitat, especially soil-specific, than animals. This may now prove to be yet more the case in hyperdiverse tropical forests than in other vegetation. Plants create the physical structure upon and within which all other life exists and depends for shelter and food. Plants are therefore the primary supporter of overall biodiversity, which is also consequently habitat-specific. First, then, our research shows the way for a more intensive habitat-specific form of silvicultural management, whether for timber or other goods, or for the conservation of limited areas of biodiverse forests. Different rainforest regimes are associated with different tree species. Global climate change can be expected to have dramatic influence on which survive and even spread, and which retreat. At a local level, soils and topography greatly influence species composition and diversity, as we've seen. There are good prospects for enhancing the abundance of particular common species or species group with particular timber or other characteristics if management is modified to suit specific habitats and species associations. Plantation design must also take local habitat into account and the lowering of risk from pests and diseases that will be achieved by mixed species plantings. Second, in the 50 years that I have spent in tropical Asia, unlogged lowland primary forest, home of the world's greatest biodiversity, has become almost entirely confined to the scattered, legally protected areas in national parks, nature reserves, and wildlife sanctuaries. Logging, by changing canopy structure and reducing the area of the mature stands where tree species diversity is concentrated, will increase and expand local extinction of the rarest species at each felling cycle. Logging is incompatible with conservation of biodiversity overall. Third, tree species also vary with habitat. As overall biodiversity is correlated with tree species diversity, conservation planners must ensure that all habitats are adequately represented in conserved areas if species diversity of plants and the many animals and microorganisms with species-specific dependencies on them are to be saved. Habitat specificity of rainforest trees has been found to be even greater than that of trees in our temperate forests. Most species occur, therefore, in nature, in archipelagos of island habitats across the landscape. That's good news for plant and overall biodiversity conservation. For them, the laws of biogeography apply at the habitat island rather than at the regional scale. Logging will reduce the number of islands, thereby certainly increasing risk of extinctions, but not on the same scale that is predicted for motile vertebrates, especially the larger birds and mammals for whom the landscape is a more homogeneous habitat. Next, plants require different conservation strategies from vertebrates. Birds and mammals are most abundant in the most productive forests. Those on fertile soils, here we have a gradient of fertility or productivity, and here a gradient of disturbance in this diagram. They're found on the most fertile soils, and especially in disturbed forest, where lush new growth is abundant. In nature, these habitats are on volcanic soils and on floodplains where trees are shallow-rooted and easily blown down. But these areas are actually quite low in tree and therefore overall biodiversity, which we found, as you saw in earlier diagrams, to be concentrated in forests of relatively low soil fertility, down here, and relatively low levels of canopy disturbance. Thus, selective logging, you can see at once, may actually increase numbers of many vertebrates, but lower overall biodiversity. Conservation agencies are focusing their attention on saving large contiguous areas inland. And this is a diagram from World Wildlife Fund. Big areas inland like that here in Borneo. Much of it mountainous, much of it logged, because they offer the greatest hope for the threatened large rainforest vertebrates, the orang-utan, rhinoceros, tiger, elephant, and hornbills. But that will not save biodiversity overall, which has already retreated to a few small isolated reserves, small forests, often near the coast. This is Kinabalu here. These small coastal forests, and down here as well. And these are in the lowlands, often inadequately policed and often gnawed by illegal felling and over-harvesting of other products, and of course, over-hunting. Science has defined the priorities, as you can see, and identifies the principles for sound management now. It is now for the conservationists and their public supporters to act. The history of forest decline in Asia since the arrival of global markets has shown that those cultural traditions with a heritage of spiritual respect for nature have best sustained their resources. Science and the law proves insufficient. We must come to appreciate that science deepens, not weakens, our mystical reverence for nature. Although recent research is revealing in many organisms, including tropical trees, that many of our current species diverged only some 3 to 7 million years ago, most are still more than 20 times as old as our own species. Their extinction at our hands will not, we can be almost certain, be repaired within the lifespan of humankind. Japan, as the most forested of the advanced industrialized nations of high population density, serves as a magnificent example to other nations. Japan must lead in sharing that example with the less fortunate nations which hold the irreplaceable genetic library that is the tropical rainforest. But in our world, we must also ask, who should be paying for biodiversity? The poor country owners who have more profitable uses for their land, or the principal beneficiaries? They are us. Thank you.
Ashton 博士、どうもありがとうございました。Thank you, Dr. Ashton. これで3人の受賞者の方の講演を終わります。Professor Fell, Professor Dr. Grunberg, and Dr. Ashton, thank you again for your commemorative lectures. 皆様、3人の受賞者の方々にもう一度大きな拍手をお願いいたします。